Everyone, and welcome to day 2 of the Forest Growers Research Conference for 2020. I'm Ian Hinton. I'm the chair of the FGLT Forest Research Committee, and in my day job I'm a senior manager with Timberlands Limited. If you're anything like me and you need a second cup of coffee to get yourself going, now's the time to grab that. In the meantime, I have the pleasure of introducing the first 2 sessions of this second day. The first session will be chaired by Paul Adams from Rainier, focusing on health and vitality of our forests, and the second session will be run by John Moore, focusing on how we can improve productivity and profitability of our forests. Before we get into the detail of the day's proceedings, I'd like to deviate slightly. Most of you know Russell Dale, and most, but perhaps not all, will know that Russell is finally, as his wife would say, retiring at some point close to the end of this year. Russell, I've known him for 30 years. During that time, he's been a senior manager in the Forest Service, in Forestry Corporation, Fletcher Challenge Forests, Timberlands Limited, and Carter Holt Harvey. And that was all before he joined FOA as the Forestry Research Manager in 2007. Since 2007, Russell has worked with all of us, worked very hard to change the face of research. He's used his unique skills to bring together industry at all levels. He's able to work with the doers on the ground, also with CEOs. He's created fantastic relationships with science, so that scientists, science administrators, not just with science but across other sectors, and importantly with government. And it's that unique set of skills and the way, Russell, that you have applied those unique set of skills that is a huge part of why we've been so successful in recent times with research programs that are providing great opportunity and improvement to our current forest industry. So Russell, thank you very much, and we hope you enjoy your retirement as much as you enjoyed working with all of us. Cheers. Okay, well, back to the agenda. Just a note that the comments that I'm about to share with you are my own views. Some of you will share them, some of you won't. But I think what's important is that we recognise that the past is a good predictor of the past. It's not necessarily an accurate predictor of what the world may look like going forward for forest produce. No doubt, though, in my mind that there is a real opportunity for forestry and for forest fibre. Now I say fibre as opposed to saw lumber, which tends to be the backbone of the New Zealand industry. There's evidence out there that the world will need 3 times the amount of fibre that it currently has available, and that's a real opportunity for us. What's not so well understood is the form of that fibre, and the examples I've got in the next couple of pictures. The first one, that's an iPhone 6, so not very big, and that is the feedstock into a rather large mouldings plant in Brazil. The fibre supply to them is 16-year-old Pinus tator, and they use the whole of that resource. You can see in the raw sawn, the green sawn lumber, that they're producing mouldings out of the core wood, the juvenile core, and they do have substantial issues with distortion on drying. The next slide is taken from South Australia. It's the South Australians utilising small thinnings down to 5 centimetre SED. Here in New Zealand, I don't think there's anybody who's utilising logs of that small dimension. Cost is another issue. This photo is a forestry supervisor visiting his crews. He's using a motorbike as opposed to the next slide, which is a four-wheel drive, stuck in the supposed sand forests of South Australia. And I can assure you it was a bloke driving, not the woman who was digging them out. So how do we lift productivity? And I'm not telling you anything you don't already know, but it's that mix of genetics, nutrition. And when I say nutrition, I mean soils as well as mineral fertilizers. It's also silviculture, so it's how we bring all of those together. And we cannot forget the customer. So wood quality is paramount as well. The agricultural world has been working with the genome for a long time, and we're just starting to get going. And if we're able to double the annual lift in productivity from genetics, that will make a huge impact on the quality and productivity of our sites. Equally with nutrition. And again, as I said earlier, I don't think of nutrition as just mineral nitrogen or mineral fertilizers. It's about understanding the soil biome. You will see in a subsequent talk that Zion is starting to do some pretty exciting work in this area, and I think we all look forward to the results of that. Examples of other work going on in the productivity space, it's work around land prep and how that impacts on productivity, work with Despot, understanding the internal features of different genomes. And of course you can link that either into customer strategies or back into your genetic deployment processes. Measurement of the forest, you've got hover map, you've got LiDAR, and also nutrient flows. What happens when you fertilize a stand with mineral fertilizer? Does it go into the trees? Does it volatilize? Or does it leach? And those are all key questions that need to be asked, and work is going on in those areas. And finally, in this section, silviculture, get it right. In the first slide, you maximize carrying capacity. Get it wrong, you have disease, you have wind throw, and the like. The last slide is mixed land use in Brazil. The farmer here finds it more profitable to run forestry, in this case eucalyptus, cropping and cattle on his farm. And as I said earlier, the world, in my view, will not be the same going forward as it has been in the past. We are going to get more and more pests and diseases. Climate change is certainly driving some of that. First slide here is from Germany, and that's the effect of the bark beetle. Fire's the next issue. This particular photo is from South Australia. It took 15 years to grow and 20 minutes to burn down. The next one is just above the Yosemite Valley. Climate change has brought about hot, dry summers. Bark beetle has come down from Canada as a result of climate change, and ideal conditions for fires. And then finally here, spraying. Spraying is a mainstay for New Zealand forestry, particularly in the establishment phases. There's a lot of pressure now, and I'm unsure that we'll actually be able to continue with those practices. Technology is going to become a driver of how we work in the future. Remotely operated machines, real health and safety opportunity there. The Aerosonde, that's a pilotless drone, the ability to stay up in the air for 7 to 10 hours at a time, measuring the forest, and of course driverless as opposed to rudderless trucks will make an appearance at some point in our industry. In the future, we won't be managing our forests just for fibre. We will be managing them for other purposes. And finally, our people, we need to be thinking very carefully and clearly about the type of skills that we're going to require to drive ourselves forward in the future. Okay, well, thank you for listening. I hope you found the conference stimulating and thought-provoking. I think we have a really exciting future ahead of us, and over to you Paul and John. Cheers.
Good morning and welcome to Day 2, Session 1, Topic 1 of the Forest Growers Research Conference. As introduced by Ian, Session 1 is about the forest industry's ability to respond to future challenges. This first topic is about managing the health and vitality of our forests. My name is Paul Adams and I am the Forest Research Manager at Matariki Forests, based in Auckland. I'm also the Chair of the FOA Forest Biosecurity Committee. The ongoing health and vitality of our forests is key because we depend on forests for wood and wood-based products, along with a wide range of essential ecosystem services, but also cultural and spiritual connections. Our ability to maintain and improve the health and vitality of our forests into the future will be a key driver for ensuring that our forests continue to be productive and continue to provide these vital products and services. We are all aware of the increasing pressures and challenges on our forests and the increasing risks from things like fire, wind, drought, floods, and pests and diseases. And many of these challenges and pressures are brought on by climate change. As an industry, we're being proactive in our efforts to improve the health and vitality of our forests through science, innovation, and technology. And this morning we have 6 presentations about this. The first presentation is about fire and how to predict where and when it will happen, its behaviour, and the technology that is being used to develop this knowledge. This presentation is by Grant Pierce, who is a senior fire scientist within Science Rural Fire Research Group. He has been studying rural fires for over 28 years and is the science leader for the current MBIE Extreme Fire Program. Today, Grant will be presenting an update on the research being undertaken within this program. Following Grant, there will be 3 presentations about pest and disease management and how to detect and manage their impacts on forest health. Rebecca McDougall will lead off with a presentation about protecting plantation forests from pests. Rebecca is a microbiologist specialising in forest pathology and forest diagnostics, characterising pathogen populations and disease management. Next, we'll hear from Steve Pawson, who is the Senior Lecturer in Forest Health and Biosecurity at the School of Forestry. His talk is about pest and disease management in durable eucalypts. One of Steve's interests is developing new technologies for active surveillance, and a great example of this is the new mobile app called Find a Pest. The 4th presentation will be by Jessica Kerr about the Picara of Protection Research Program at SCIOM. Jessica works in the entomology team and is well regarded as a technical expert in entomology and pest management. The Picara of Protection Research Program has multiple areas of research, and Jess will be talking about 5 of these projects. The last 2 presentations are directed at forest nutrition. Mike Watt from SCIOM will discuss emerging remote sensing technologies, particularly hyperspectral imagery for nutrient management. Michael specialises in remote sensing and models to characterise the growth and wood quality of the forest resource. He has published over 160 refereed papers around these and other topics within forestry, and he is the co-leader of the Remote Sensing Cluster Group. The final presentation about the health and vitality of our forests is by Loretta Garrett, a science scientist who specialises in forest, soil sustainability and carbon. Her research encompasses the long-term sustainability of planted forests over multiple rotations. Her presentation this morning is about the use of high throughput, low cost soil and foliage analysis. We trust that you will enjoy these 6 presentations. There will be a Q&A at the end, so please forward on your questions so they can be addressed at the right time. Thank you.
Hi, I'm Grant Pearce. I'm a senior scientist with the Scion Rural Fire Research Group, and I lead the extreme fire program that we'll be talking about today. So I'll just start my presentation. All right, so I'm going to be talking about the extreme fire program. So the extreme fire program is a 5-year program, and we're just currently entering the last year of that program. So we finish in September next year, and we're currently looking at rebidding the program for what might come following the end of this program. But I'd like to talk about 3 aspects of the program today: the extreme fire behaviour research burns, the real-time fire monitoring tools, and the extreme fire prevention sensor technologies. So the program is funded by NBIE, but we receive co-funding from the Forest Growers Levy, from other partners, Fire and Emergency New Zealand, Department of Conservation, and also New Zealand Defence. So the first area I'd like to talk about are the research burns. We're testing a new fire spread theory developed by the US Forest Service's Missoula Fire Sciences Lab, based on a series of lab fire experiments. And those studies have shown that radiation on its own is insufficient to ignite fuels, especially those loosely arranged fine fuels in which fires start and spread. And their theory suggests that convection is much more important than previously thought, and perhaps as important, if not more important, than radiation in transferring that heat ahead of the fire front to the unburned fuels. And convection acts to set up a series of up and down drafts above the fire front, where the heated air rises, but then as it cools, comes back down and pushes down into the flame front, pushing the flames out into the fuels ahead of the fire. So those fuels are then preheated through radiation, but also this convected heat and through direct flame contact. If proven, this new theory could fundamentally change fire science globally. It would change the way we teach fire science and heat transfer to firefighters, and ultimately result in more accurate fire spread prediction models. Then in turn, that will lead to better decisions being made during firefighting operations, and ultimately to safer firefighters and communities. So the study aims to conduct burn experiments in 3 different fuel types: cereal crop stubble, where we conducted burns in 2018 and 2019; gorse scrub fuels, where we recently completed burns in March this year; and eventually moving into, we hope, standing wilding conifers over the next couple of years of the program. And here we aim to measure these heat transfer characteristics and other fire properties, including fire spread rates, flame lengths and intensities, for example, with the ultimate aim of improving understanding of extreme fire behaviour and fire spread. These research burns are a major international collaboration, and as well as ourselves and the Missoula team, we are also collaborating with the University of Canterbury's Geography Department, the San Jose State University's Fire Weather Research Lab, the University of New South Wales from the fire modelling standpoint, and we've also used the expertise of the Scion UAV team. During these burns, we deploy over $2.5 million of equipment in the form of high-resolution thermal cameras, LiDAR, drones, wind anemometers and other sensors. And each of the collaborators bring some of the specialist equipment and expertise to the program that enables us to do research that we would not otherwise have been able to do here in New Zealand on our own. So the gorse scrub research burns were just completed in March this year. We completed 6 burn experiments in these more complex, non-uniform scrub fuels. Each time we were burning a 4-hectare research burn block, with 2 burns being conducted per day, with 3 burn days over the 7 days that the research was conducted. But prior to that, we required 3 further days to complete the burnouts to secure the site, to prevent escapes. And the burns involved the 30 research personnel, but more importantly, also the support of over 35 to 40 foreign emergency, dock and other staff and suppression equipment. And we're very grateful for that support. So this is some footage from one of our in-fire cameras that shows the flame front approaching. And here you can see that we got extreme fire behaviour as we'd hoped, as the burns burn through the sensor arrays that were deployed within the blocks. Hopefully you can also get a sense for the noise and the heat that's generated through these burns. So looking at some of the results to date, these gorse burns were hot and definitely hotter than the previous stubble burns that we'd experienced. The spread rates were in the order of 3 to 4 kilometres per hour, with all of the burns being over within less than 4 minutes from ignition. Flames in the order of 8 to 10 metres high on average, but with peaks in excess of 20 metres, and temperatures measured using some of our sensors at over 1500 degrees Celsius, which was enough to damage some of our research equipment. Although thankfully we were still able to capture the data out of those damaged sensors despite the heating that had occurred. So the new purpose designed instrumentation was successfully tested and utilised, and we were able to capture excellent data for the 2 different fuel types we've burned in so far. And from the results to date, they're showing that the new fire spread theory looks to be valid, at least in these 2 gorse and stubble fuels that we've burned in to date, but it looks very promising as we move forward. We've also been able to develop new analysis methods for fire spread rate and hot air parcel movement above the flame front from the video imagery. And these burn experiments are the first known attempts to quantify the turbulence generated by these flame fronts across all the different scales, from the background atmosphere to above the fire to within the flame front, using the specialist equipment we were able to obtain. And a new prototype fire spread model is currently under development that will be further testing using the data collected from the burn experiments. So the next steps are to move on to conducting burns in standing wilding conifers, and we have a site near Lake Pukaki in the Mackenzie Basin, where here we have 10 to 12 metre tall dense wilding stands, where we hope to now burn in 2021-2022. All of that was put into a state of turmoil recently with a fire late last month, but thankfully that missed our research blocks, and hopefully we can still burn in a year's time. All right, so the second project I want to talk about is the real-time fire tools. So here we're looking to develop an integrated tool suite that combines a series of separate tools for fire detection, fire spread and growth, and for the dispersal of smoke produced by the fire. So this system will be able to detect when a new fire has occurred, and from that location and the fuel types it's burning in, use the Prometheus fire growth model to predict how big the fire is, what's getting consumed in the way of fuels, and what's the rate of spread and of that fuel consumption, which then is input into the smoke model, which then predicts the emissions that are produced by the fire and how high the smoke is going and where downwind that fire is, that smoke is likely to end up. So a prototype web interface has been developed, and we're going to test the beta version of this integrated model over the coming fire season. So to date, we've been able to successfully develop the individual components and link them together. And now we want to test the full integrated suite over the coming fire season. And we're also in discussion with Fire and Emergency New Zealand regarding how we take this system forward and implement it operationally and use that information in real time to support fire decision making. So as I said, the system combines three different components. The fire detection component. So here, these are the satellite fire hotspot detections, in this case from the MODIS satellite, which are then used to generate fire spread from the Prometheus fire growth model, which in turn is then used to generate the smoke and where that smoke is likely to go. And so the interface also allows us to be able to download that information so that we can then overlay it over the likes of Google Earth imagery to show the fire spread in this case from the Prometheus model, but also the smoke. And we can animate that into a series of short video clips showing fire and smoke spread as well. All right, so the third area I want to talk about is the development of a fire alerting sensor to warn of the approach of fire. And this is a simple temperature measurement made via a thermocouple that's connected via a communications package that sends alerts when a temperature threshold is exceeded. So in this case, we've been using temperatures above 65 degrees Celsius. So this sensor sends the location and the temperature data, and the frequency of that sending of the data increases as the temperature increases. The system has been tested over satellite and radio communications, and it can be linked to the internet or made accessible via a smartphone application that alerts somebody monitoring that to a high temperature reading from one of the sensors. These sensors have been successfully trialled in our stubble research burns, but also in our more extreme Rakaia Gorge Gorse burns this last year, where they showed the approach of a flame front and were able to survive the temperatures in excess of 1,000 degrees C when properly insulated. So the aim here is that the sensor can be used around the boundaries of a prescribed burn to indicate where there might have been a breach or escape, or they could be used as a quick deploy sensor within the forest, or dropped from the air above a fire to indicate the spread of a fire as well. We've also been able to link the sensor to the operation of fire suppression systems, and in this case to automatically switch on a sprinkler when the threshold temperature was exceeded. And here it was able to turn on the sprinkler via both electric water valves if the pump was already running, or if you had an electric start pump by switching on the pump itself. And we successfully demonstrated the use of the sensor in this way at a workshop that we held in Northland in August last year. Work's also investigating use of a modified sensor to monitor the internal temperature within skid site piles. And here the aim is to reduce the occurrence of costly spontaneous combustion fires by monitoring pile temperature remotely and providing an alert when, again, a threshold temperature is reached. And that links to some other work that we're doing within the program and for Fire and Emergency New Zealand around the factors contributing to the occurrence of skid site fires. And the report is due on that very soon. I just want to finish by highlighting some of the tech transfer mechanisms we're using to get this information out. So hopefully you're aware of the research web blog that's now been running since June 2018. You can subscribe to the web blog via the Rural Fire Research website. And here we provide brief updates on research activities and new publications, etc. But also to the new YouTube video channel that we've created. So if you go to YouTube and search Rural Fire Research, you'll find the channel there. And here we have a selection of videos from research burns, other research activities and presentations. And to date, we have around 24 videos uploaded for viewing. So thank you very much for the opportunity to update you on the progress of the Rural Fire Research and of the Extreme Fire program in particular. If you want more information, you can get information from the Rural Fire Research website, which is, the link is listed here, and also through a link from the Forest Growers Research website as well.
Hi, my name is Rebecca McDougall. Today I'm going to be talking about some work we've been doing about protecting our forests from pests and diseases. Firstly, I'm going to talk about two diseases causing major forest impacts overseas. These are Phytophthora ramorum and Lecanosticta acicola. Secondly, I'm going to discuss two diseases already present in New Zealand, but that are currently behaving a little bit differently. And then I'm going to introduce Daryl Heron and some of the work that he has done at FEBI and also in his new role with us at Scion. So firstly, can Phytophthora ramorum infect pine logs? We know that Phytophthora ramorum can infect Pinus radiata and Douglas fir, and that it is considered a major threat to many ecosystem types due to its broad host range and impact on host survival. The aim of this study was to determine if Phytophthora ramorum can infect logs post-harvest. This would provide assurances to international trade partners in the case that Phytophthora ramorum ever established in New Zealand. This work was completed in Oregon, USA, and 3 tree species were used: Pinus radiata, Douglas fir, and tan oak as a positive control. 3 log pieces of each were placed in 4 different sites in Oregon. And you can see in the photograph in the bottom right-hand corner a picture of how those logs were set up. The logs were left in the forest for natural infection, and bait buckets were used to detect inoculum present. The results of this study were that from 36 log pieces across 4 sites, only 2 tan oak log pieces tested positive for Phytophthora ramorum. Some lesions were observed on the Pinus radiata logs. However, Phytophthora ramorum could not be isolated from those. The conclusion was that Pinus radiata logs are not susceptible to Phytophthora ramorum colonization, and this demonstrates that the risk would be low for post-harvest logs. Next, I'm going to talk about Lecanosticta acicola in Spain. Lecanosticta acicola is the causal agent of brown spot needle blight. This fungus is found in the Northern Hemisphere, but also in Central America and Colombia. We recently visited Spain in April 2019 to observe this disease and to build connections. This was a great opportunity to see the disease firsthand and the impact it is having in forests there. It was also good to make connections with Spanish researchers and to become more familiar with the disease. Ceridium. Ceridium has always been associated with Cupressus species in New Zealand, but we have now recently detected Ceridium species on Pinus pinasta and Pinus radiata. On these species, we have observed small cankers on the branches and the twigs, and the pathogens have been isolated from needles. It is not known to cause disease on pine anywhere else in the world, and Scion pathologists are currently investigating the taxonomy of the isolates from the 3 locations. We have also conducted a field visit to observe trees in the field, and analysis of those samples from the visit is underway now. Nectria flute canker. Nectria has been present in New Zealand for a while now, and it is caused by Nectria foucaultiana, formerly called Neonectria or Nectria foucaultiana. This year, the pathogen was discovered north of its previously known boundary. In a recent Forest Health News article of August 2020, the map on the right-hand side of the slide was presented, showing the results of a Nectria flute canker survey from 2005 to 2008, but also showing the new finds in 2020 above Christchurch. Is this pathogen on the move, and why? Since the reclassification and during our diagnostic analysis, it became quite apparent that more work is needed. We need to understand the species and the taxonomy of the populations here in New Zealand and relative to the overseas populations. Are there other factors responsible here? Is a changing climate, such as climate warming, or genetic change in the pathogen responsible for this? This is yet to be understood. So lastly, I just want to introduce Daryl Heron. Daryl is currently based at FEBI at the University of Pretoria, but is also working for us here at Scion. Daryl started working for us during lockdown for COVID-19 back in April 2020, and at the moment he's currently completing a PhD on Fusarium at FABI. This PhD is looking at Fusarium succinatum, the causal agent of pitch canker, that's also associated with grasses in South Africa. He has over 12 years of experience working in diagnostics and extension within the Tree Protection Cooperative Program based at FABI and supporting commercial forestry in South Africa. At Scion, Daryl is a forest pathologist and the assistant research leader for diagnostics and emerging threats. Since he's been working for Scion and working remotely from South Africa, he's been completing some review documents for us and for industry as well. Firstly, he's looked at a review to the risks of the risks to pine and Douglas fir. This review showed that the biosecurity risk is increasing for our forests, but it's balanced by advances in technology. Daryl's recommended that some level of diversification would be prudent, and maintaining genetic diversity is also important. He's also suggested that the most appropriate form of diversification is yet to be determined. This slide shows some work that Daryl has been doing as part of his PhD in South Africa. This is really interesting work on Fusarium succinatum origins and the variation in strain aggressiveness, and highlights the quarantine risk of these pathogens. We know that there are problems with name-based risk assessments and regulation decisions made just on a name. Where possible, information such as alternate hosts and population structure should be considered when making decisions about how to prepare and manage an important pest or pathogen. This work involved a pathogenicity trial on more than 50 strains of Fusarium succinatum from various countries around the world, including South Africa, Spain, Colombia, and the US. The trial showed a wide spectrum of virulence between strains within countries and between countries. This variation is important to note for countries such as South Africa that have developed pine breeding programs challenged with local strains of the pathogen. Breed material might be susceptible to more virulent strains of Fusarium succinatum from other places. Identifying centres of origin and countries with virulent strains can help focus quarantine efforts and better prepare breeding programs for new strains that may enter a country. We're really looking forward to Daryl being able to join us in New Zealand once he has finished his PhD and is able to come to New Zealand. Lastly, I'd like to make some acknowledgements. Firstly, we'd like to acknowledge the Forest Growers and the Forest Owners Association for supporting the work that we do. We'd like to acknowledge the Oregon State University, Jared Lebaldis and his team for the work on Phytophthora ramorum. We'd like to acknowledge NICA in Spain for their support in visiting Spain and the forests there. And we'd also like to acknowledge SPS Biosecurity for the work that they've done around the Nectria and Ceridium detections as well. Thank you very much for listening.
The time to stop runaway global warming is running out. Fast-growing forest trees are the most effective way at this time for us to fight climate change. They suck carbon from the atmosphere. That's a big environment plus, and there's more jobs and income if you grow trees than farming sheep and beef. That's why so many smart farmers are so bullish, planting out trees and loving our forests.
Kia ora koutou, ko Steve Paulson toko ingoa, e mahi ana au ki te kura ngāhere, ki te whare wānanga o Waitaha. Kia ora, Steve Paulson here from the School of Forestry at the University of Canterbury. Many of you probably know me from my past role at Scion, leading the entomology team. Now I'm here at the University of Canterbury working in forest health and biosecurity. So today I'm going to be talking about some of the pests of dryland eucalyptus. So the New Zealand Dryland Forest Initiative was formalised in 2008, although it started a little bit earlier than that. And the focus of the program is to develop a naturally durable source of timber to replace CCA-treated pine. So the program itself is investigating a number of species, but there's 5 in particular that they're taking forward in their breeding program. So what we know about eucalyptus is that, yeah, it's got this naturally durable timber, but also insects are somewhat attracted to eucalypts. And we've been receiving quite a few of them from our friends in Australia, which is the, shall we say, centre of diversity for eucalyptus, but it's also the centre of diversity for eucalyptus pests. And we're just downwind, so not only do we get the smoke from their bushfires, but we also occasionally accumulate insects from their forests that blow over or hitchhike with us on international trade when we're moving goods between Australia and New Zealand. So we've been receiving insects from Australia for quite some time now. Some of the earliest invasive forest insects came from Australia and fed on eucalypts, things like Phoracantha, semipunctata that arrived as early as the 1860s. And we've been steadily accumulating the odd thing ever since. So the situation today is that there's 2 really key defoliating species that are a real big problem for trying to expand the eucalyptus growing industry to a larger variety of species, but also to protect the fairly sizable Eucalyptus nitens plantations. So these 2 species are both pyropsene beetles, and one has been here for over 100 years, Pyropsis charybdis, and Tony Withers has spent most of her career looking at biocontrol options for Pyropsis charybdis, otherwise known as the Eucalyptus tortoise beetle. But more recently, another significant defoliator that's possibly going to be worse than Pyropsis charybdis arrived in 2016. And this people probably know as the Eucalyptus variegated beetle. It was known by the scientific name Pyropsis vericollis, but when looking into that particular species in more detail, they have recently found through molecular studies that it's actually a complex of different species, and the one that we have is a thing called Pyropsis cloelia. So a key part of what Leslie's trying to do for her PhD is to examine whether or not some of these eucalyptus species that form part of the Drylands Initiative exhibit what we call resistance or tolerance. So there's quite a big difference between these 2 things. So resistance is where the herbivore doesn't really want to eat the plant. So it doesn't taste good. So either there's nasty compounds in the leaves, or there's some sort of tough fibrous nature to the leaves that the insect doesn't want to feed on that particular plant. The alternative option is that the plant has developed strategies to cope with the feeding. So it's a more stable coevolution than resistance. Resistance is a bit of an arms race. One develops a defense, the other one tries to overcome the defense, then it needs a new defense. But in terms of tolerance, the plant has learned to live with it. It doesn't mind having something having a bit of a nibble on its leaves, and it continues to be productive, put on height, put on DBH. And so those are the kind of things that we're looking for when we're trying to make breeding decisions around protecting these eucalyptus species from herbivores. So as we go on, we'll be talking about resistance and tolerance. So I mentioned earlier that there's 5 key species that the Drylands Initiative is interested in as part of their main breeding program. And these fall into 2 subgenera of the eucalyptus. And you may have heard of these names. There's the Symphomertus and the Monocalyptus. Now most of these are symphomertus, and unfortunately what that means is the insects think they're like chocolate. So insects in general prefer the symphomertus eucalyptus than they do the monocalypts. So the colours that I've got on the presentation here are quite important. So the red ones that I show you on the next slide are the symphomerts, and the blue ones are the monocalypts. So let's take a look at some of the previous work that's been done by Hui Min Lee, who finished her PhD a few years ago, and she looked for evidence of resistance. And so she examined a range of species using a fairly coarse measure of how much the herbivores were feeding on the trees. So damage levels A, B, C, and D. So D, the darker the colour, the more damage that these have. And what you can see is that by and large, the more heavily damaged species were the Symphomertus. The one that's probably the exception there is Eucalyptus cladocalyx. What we don't know is whether this resistance is heritable, so can it be passed on genetically as part of a breeding program? That would be a latter topic to be looked at. But what we can see is that there's 3 species, cladocalyx, macrorhyncha and globoidea, that seem to be the ones that are most resistant to the browse of these 2 pyropsines. So Leslie is taking the work of Huimin further, and she's using an index of defoliation called the crown damage index. So higher values of the crown damage index mean greater defoliation. And one of the really pleasing things that we can see in the very early stages of Leslie's PhD is that she's seen the same results that Huimin did. So Huimin was working in the North Island largely, Hawke's Bay, and these results are from the South Island, from the Marlborough region. And we're seeing the same 3 species, the macrorhyncha, the cladocalyx and the globoidea, having lower levels of defoliation than all of the other Symphomerts. Cladocalyx is still that standout oddball that seems to have low defoliation, despite being one of these Symphomertus species. So there's quite a large range of damage between species, which suggests that there is some opportunities to select for species that are more resistant to the browse by these particular pyropsines. But I guess the big thing that we want to know is how is this affecting growth of these Eucalyptus species? So this is very preliminary data for Leslie. So she's in the first year of her PhD and she's taken measures of defoliation, which I showed you on the previous graph. And this is the first measure that she has of the growth of these trees, so the DBH of these trees at that point in time. Leslie's subsequently going to go back and she's going to resample these plots, again looking at defoliation, but again measuring the DBH. So we can see what the growth increment will be over the course of 1 year relative to the defoliation that they suffered. And what we can see at these particular sites, Dillon and Leeseman, so the left and the right columns in each little pair is Dillon and Leeseman. They were established at different times, so we would expect the DBHs between the 2 sites to be quite different. But what we can see is that those species that were less defoliated, we're seeing on average a greater DBH. Those are the horizontal lines that I've put, sort of just a rough average in there. And there are these outliers that are much greater than the average. So what we need to do now is try and figure out, is that an environmental effect that's causing that? Or is that something about the genetics of those species that we can work with that might be repeatable in other locations? So are there some species that are more resistant? And over the years, as Leslie collects more information about growth as it occurs with the defoliation, we'll get an idea if that is expressing tolerance as well. So one of the really critical things that is going to allow us to expand on this work is to find rapid ways to assess defoliation of eucalyptus on a large scale, so a whole site basis. So to date, we've been reliant on things like pest counting, tree grading, which is Huimin's work, and the crown damage index that Leslie's been using. But these are very time consuming. They require an individual person to go and assess every particular tree, potentially cut shoots off those trees, bring them down and assess them individually. You can't do that at scale, and so therefore we can't support big breeding decisions that need to be made at scale in terms of assessing a whole pile of different families and clones across multiple sites to sort of examine if there's side effects. So the way forward that we want to go is we want to go aerial. So let's get some eyes in the sky and use some imagery, be it RGB imagery or LiDAR. Leslie's going to be working as one of the projects in her PhD is to evaluate, can we come up with a method to evaluate the level of defoliation of these eucalyptus in a repeatable manner that we can then correlate with the levels of defoliation that Leslie is observing on the ground. So the idea is to get geospatial data or remote sensing data that we can then correlate with the actual observations that we make on the ground of the levels of defoliation. And that'll be a really useful tool going forward that can not only be used to make, shall we say, decisions for breeding, but also could be for management decisions in eucalyptus plantations, i.e. have things got out of hand and do we need to implement a spray perhaps to knock the population back? So talking about future plans, we actually have 2 PhDs on offer at the moment. So if you know any bright young students that want to come and work with me and Tony Withers, one of them is going to be on the Eucalyptus variegated beetle, so this Peropsisterna cloalia, and we're going to be looking at how some of the generalist predators, like the Clearbora ladybird beetle in the image there, might help control the pyropsene beetles. So how might we be able to make life better for the ladybird such that it can have a good go at these pyropsenes? And one of the things that we're quite interested in looking at is having mixed stands where we have a number of different tree species in there that get cut out during the thinning process, but these species will help the ladybird beetle thrive and hopefully exert more control, thus resulting in better outcomes for the main crop species of the eucalyptus. So as I mentioned earlier, the pyropsene beetles are the key defoliators of eucalyptus in New Zealand, and Tony, Withers at Zion and others have been working a lot on parasitoids of these species for other reasons other than the dryland eucalyptus project. Now what they've found is that one of those particular parasitoids, Anogra, is actually pretty good at parasitizing the new beetle Prometheus cloalia, the Eucalyptus variegated beetle. Almost 50% of the eggs in one study in the North Island where they were looking at this were parasitized by Anogra. And that's a great result, which means that we can look forward to hopefully having some good control by this species of variegated beetle. Now the unfortunate thing is the other parasitoid, Neopolycystis, it also attacks the new Prometheus cloalia, but unfortunately it doesn't result in viable offspring. So it's a bit of a double whammy in that we're not actually controlling the cloalia, but also they're kind of wasted effort by these little female parasitoids that could otherwise be stinging some Prometheus charybdis eggs and dealing with the other species. So many of you are probably aware that the EPA recently approved the release of Aedia daenerys. Now this is a new parasitoid, not currently present in New Zealand, and Tony and others at Zion have been working very hard for many years to get all the host specificity data, and it was recently approved. So over last Christmas they were working hard trying to breed up many individuals from a cohort that they brought over from Tasmania, but unfortunately they couldn't release them into New Zealand outside of Zion's containment facility because it was found after they were all bred up that they were contaminated by Microsporidia. So big disappointment for the team, but they're going to be trying again this coming December. So Aedia daenerys is another parasitoid of Prometheus charybdis. So that's great news for controlling Prometheus charybdis, but it doesn't attack cloalia. Now, as part of the release process or part of the research that's gone into Aedia daenerys, what we found, or what an international team that Tony's been part of has found, is that actually the Aedia daenerys is part of a species complex. And there's actually 3 or 4 species in there, and one of them, Aedia and Lecii, does go after Prometheus cloalia. However, to get permission to release that in New Zealand, we have to go through the whole host specificity testing. Now, that's quite a large piece of work, 2 or 3 years worth of work, but I think it's probably worthwhile that the industry starts thinking about that if they want to have an expanded eucalyptus industry, because it's likely, or what we're seeing so far in the field, is that Prometheus cloalia is going to be as bad as charybdis or potentially worse. So currently we don't have any specific focused parasitoids on that species, so it'll be good to think about Aedia and Lecii for the future. So I guess the key message is that eucalyptus is like chocolate. Everyone kind of likes it, but they like some flavours more than others. And that's what we're finding with these pyropsene beetles. None of the species are 100% resistant to browsing by these species, but definitely some species seem to be more resistant than others at this point in time. However, because we're not seeing really high levels of resistance by any of these species, we really want to understand this tolerance. So how tolerant are they of a little browse such that they can continue being productive and putting on the DBH and the height? So that'll be a key focus of us going forward, is understanding tolerance. And for us to make the biggest leap forward that we can in understanding tolerance means that we have to be able to assess defoliation at a really big scale. We need to be able to do it at the trial scale, so we can be looking at multiple species, multiple families and clones all at the one time. So that'll allow us to understand the most around how tolerance is happening and what site effects are. The other thing that goes alongside breeding for resistance and tolerance, obviously, is the biological control. So how can we exert top-down control to reduce populations? We know we have some parasitoids that are there already. We know we have some generalist predators like the Clearbora ladybird beetle. So how can we maximize those particular parasitoids and predators to control populations? And also, do we need to start thinking about new parasitoids, as I said, Edia and Lecii, and ensuring, of course, that we get Edia daenerys here and well established in New Zealand. So that's it from me, a little bit of a roundup of stuff that's been happening in the past, some of the stuff that's happening now, and where we plan on going in the future. So if you have any questions, feel free to ask them during question time or drop me a line at the email on the screen.
Hello, my name is Jessica Kerr and I'm from Scion. Today I'm going to talk to you about the Picora Protection Research Program. Picora Protection Research Program was a multi-year MBIE research program that ran from 2015 to 2018 and will continue with Scion core funding until 2021. The aim of the program is to avoid future long-term pest management costs in urban, production and natural environments, developing new knowledge and tools in a layered approach that combines pathway risk assessment, surveillance and efficient early detection, new models and tools for eradication, and social and cultural co-design. The Picora Protection Research Program has multiple areas of research, and today I'm going to give you a summary of 5. Firstly, insect cycle surveillance. During an insect incursion, speed of response is crucial. The faster the response, the greater the chance of success. Our research aims to actively find an insect incursion through a targeted biosecurity response tool. Many insects use chemical odours called pheromones to communicate. We have found a way to help us snoop in on the system and find them. Over the past 4 years, Scion has developed a portable electroantennogram, a lightweight tool utilising a male insect to track the pheromone of a female. The male insect is mounted in a device which can then be used for handheld active searching or placed on a UAV. In 2019, we completed 2 successful field trials in Canada with Natural Resources Canada's Great Lakes Research Centre, with key results showing gypsy moths could consistently detect insect pheromone within a wind tunnel at 30 metres and the field at 130 metres. The male moth's response is visible in the graph with the yellow boxes. We collected 676 recordings from 23 moths, a very large and complex data set that currently needs to be manually reviewed. To help improve and speed up this process, we're moving towards an automated system, and the project has been scoping a range of machine learning options for processing EAG data and delivering a prototype for gypsy moth to support our international collaboration. The machine learning algorithm picked for this type of data is a deep learning model. The model requires a training library of data so that it understands what is and isn't a response peak. We started with laboratory data, where responses were at known time intervals. The algorithm could clearly pick up the peaks after training in the bottom graph. We then moved into wind tunnel data, which requires annotation for training, visible in orange, selecting the start and the end of a response. Then running the trained model, it highlights the predicted responses in red. After training on a small subset of 3 moths, the model is close but has missed 2 responses. To improve this accuracy, we'll continue to build up a training library over the coming year, validating the model with this summer's field data coming in from Canada. Other new surveillance technology we're investigating is the use of traps equipped with cameras for targeted surveillance of insect pests. As part of a collaboration between Scion, Ag Research and the University of Canterbury, we've designed a product that will provide new ways to inform research. For example, at ports, we're planning to use these traps during and after a vessel docks to see if hitchhikers are jumping ship. Within forests, it will be used to monitor insect flight patterns. The traps are linked with the wireless Internet of Things communication network to send back basic information about the trap's performance. The project has been working on integrating machine learning for image recognition to differentiate between target insect species. The ultimate aim is to integrate these traps in the real-time Bayesian network on flight probabilities developed by Steve Pawson and collaborators, such that the sensor network can constantly improve itself. We're developing camera traps for real-time surveillance. using machine learning to automate insect image identification. The trained image recognition model can currently differentiate between 3 forest insect species based on a small training data set. It's operating at 90% accuracy. Along the same lines of research, one of our teams has been scoping the design of a system that can implement image recognition for pre- and post-border images. The species I've highlighted for you are some of the most common found at our border. The project is aiming to work with inspectors to take an image of what is found on imported goods, applying image recognition to provide important information on different taxa of insects coming through our border. The next step for these projects is to gather training data of good quality images that can feed into the image recognition model. Watch this space because the science and technology is growing rapidly. Our next project is on sensor development for detecting plant diseases with volatile organic compounds. Volatile organic compounds, or VOCs, play crucial roles in plant defense and survival. Detection of unique VOCs emitted by plants has been suggested as a promising method to monitor fungal infections or insect infestations. Microbial emitted VOCs are presently used as markers to detect human disease, food spoilage, and mold in houses. The project aims to develop a lightweight drone-mountable pathogen sensor able to sense infections prior to visual symptoms or spore dispersal. The project hypothesizes that by monitoring VOCs emitted from trees, plants, or microbes that are markers for specific pathogen species will give us a tool that can be applied to different classes of pathogen or insect hosts at early stages of infection. Over the past year, the team has developed a VOC sensor based on carbon nanofibers and has tested the sensing performance using 2 volatile organic compounds typically produced by living organisms. The results show that the sensor can be used for VOC detection. Each peak in the graph is the sensor picking up the chemical. The team will focus their sensor development on the Phytophthora responsible for cowrie dieback in the next year. Our atmospheric scientists have been researching a relatively new area of science called atmospheric Lagrangian coherent structures. Atmospheric transport of material by wind, including pests, follow preferred pathways. Lagrangian coherent structures are a way to visualize these pathways and their behavior, giving organized trajectories of winds, often called air bridges. The purpose of this study is to understand the behavior of LCS over New Zealand to optimize biosecurity surveillance sampling networks, to predict important events, and to stretch current science to understand LCS at a regional and local scale. Key findings so far from 11 case studies of trans-Tasman insect incursions show 100% are associated with the Lagrangian coherent structures. There is also a strong correlation with LCS air bridges and trans-Tasman smoke transport from last season's Australian bushfires. For each smoke event, the map showed the shape of the smoke in the atmosphere and the correlation with the air bridge. Material that is transported in the atmosphere will eventually become concentrated into these dark structures. As we see, the smoke in the cloud that is concentrated in the structures outlined in the red dashed lines. We believe this science enables us to predict long-distance wind dispersal of pests and pathogens into New Zealand, and possibly at smaller scales. Therefore, we can target eradication campaigns to where LCS predicts pests will spread. In summary, the Pecora Protection Research Program is preventing damaging forest pests and pathogens from establishing in urban, production, and natural environments. Thank you for your support.
Our planted forest industry, challenging to be our second primary export earner. Wood's environmentally good in plant and product for building and landscapes. We're great in technology and science, selecting stronger, straighter trees, safer ways to harvest them, cleaner and earthquake-resistant ways to build. Forests, regionally vital for economic development, creating all sorts of jobs. A prime future investment for our nation. Love our forests.
Good morning, my name is Mike Watt. I'm a scientist at SCIEN and I'd like to talk to you today about the use of hyperspectral imagery within forestry. This is a very collaborative project that has involved a lot of scientists. The scientists highlighted in bold are international researchers who include Pablo Zarco Tejada, who's one of the leading hyperspectral researchers in the world. Hyperspectral imagery is predominantly used to characterize reflectance across the electromagnetic spectrum. The displayed figure shows reflectance of vegetation across the spectrum. You're no doubt familiar with the visible spectrum that includes blue, green, and red, and the peak that you can see in the bottom left-hand corner shows the higher reflectance of vegetation within the green band. This increased reflectance within this band is why vegetation appears to be green to our eyes. Hyperspectral imagery has many more bands with wavelengths above the visible spectrum. Compared to the visible bands that cover 400 to 700 nanometers, the remainder of the spectrum goes from 700 nanometers to 2,500 nanometers. These invisible bands include the near-infrared band that can be used to characterize cell structure, and the shortwave infrared bands that can be used to distinguish water content in leaf biochemicals. Hyperspectral imagery has been used for spatially describing a wide range of properties. These variables include grass cover, leaf area index, and a wide range of minerals. Within plantation forestry, the main applications that hyperspectral imagery is used for include species identification, nutritional deficiencies, water stress, and disease detection. The pixel size of these spatial representations depends on the platform that is used to collect the imagery, and typically ranges from 1 meter for UAV data to 30 meters for data collected from satellite. We have used hyperspectral imagery to detect nutrient deficiencies. Application of nitrogen and phosphorus have been found to produce significant growth responses in forest stands. However, the detection of these nutrient deficiencies using traditional methods generally involves some form of foliage collection and analysis, which is costly and time-consuming. We set up a trial to determine if hyperspectral imagery could be used to detect nutrient deficiencies. We created a range of deficiencies within 1-year-old radiata pine through providing plants with a factorial range of N and P concentrations. After a number of months, the plants started to respond to these treatments, and you can see the plants with high N and P on the left of the image, which are very dark green, and the very deficient plants, which receive no N or P, on the far right, which have very light coloured foliage. The photo on the left shows the setup we used to acquire the hyperspectral imagery. We placed the hyperspectral camera in the middle of the crossbar that sits above the conveyor belt and ran the trees underneath this camera, which produced the imagery given below. The hyperspectral camera that was used was a Spectrum FX10, which can be used to capture reflectance from 448 bands across a spectral range of 400 to 1,000 nanometers. Compared to this data, you typically get 3 to 4 bands from a multispectral sensor. This slide shows variation in the hyperspectral canopy reflectance between the 5 treatments. As you can see, there are significant differences in reflectance that are particularly evident in the magnitude of the green peak, which occurs around 550 nanometers. A greater proportion of green light is reflected by the control trees with poor nutrition at this wavelength. There are also differences in higher wavelengths between 550 to 650 nanometers. Within the invisible wavelengths, there's also a red shift in the red edge, which is the wavelength range from 680 to 750 nanometers. You can clearly see that this leftward shift in the red edge for the control treatments relative to plants subject to high nutrition treatments such as N1P1. Hyperspectral imagery can be used to derive many indices from the data. Indices typically combine wavelengths with reflectance that vary significantly between the treatment with wavelengths that are insensitive to the treatment. The Carter index is shown here to illustrate the concept. The reflectance at 695 nanometers on the numerator can be seen to differ markedly between the control and N1P1 treatments, as illustrated by the 2 upper horizontal lines. In contrast, the reflectance for both treatments at a wavelength of 420 nanometers is virtually identical. Inclusion of an invariant wavelength can be used to control for other factors that affect reflectance. Using the hyperspectral imagery collected in this experiment, we've developed strong relationships between foliage nitrogen and many indices, including the Carter index, as illustrated by the two figures shown here. We have also made robust predictions of foliage phosphorus when data was constrained to the p-limiting range. Strong relationships have also been developed between indices and photosynthesis. Changing gears a little, I would like to discuss the use of hyperspectral imagery to detect wilding pines. Wilding pines are a major economic and ecological issue in New Zealand and occupy 2 million hectares, which is larger than the plantation estate. Scion has developed an effective herbicide treatment for controlling wilding pines, and this is proactively being used by DOC. Monitoring of control is necessary to determine whether repeat herbicide applications are required, and hyperspectral imagery could provide a useful means of monitoring the efficacy of wilding control. We ran an experiment 2 years ago in which 39 Pinus contortus seedlings were treated with 2 herbicides that included the systemic herbicide triclopyr and the contact herbicide dicot. 13 of these trees were set aside as control plants. The plants were about 1.5 metre high at treatment, and one of the treated plants is pictured. Measurements were made prior to treatment and for 5 weeks following treatment. This slide shows the visual changes in herbicide damage to the trees through time, starting at week 0 at the top and going through to week 5 at the bottom. As expected, there were no changes in foliage colour for the control plants. Visual impacts from triclopyr were first evidenced 7 days after treatment and intensified over time. The visual impacts from the contact herbicide dicot were evident 1 day after treatment and strongly intensified over time. The hyperspectral index, physiological reflectance index, which is a useful predictor of photosynthesis, was extracted from the hyperspectral imagery. Changes in this index over time for the 3 treatments are shown in the figure on the right. The key finding from this analysis was that physiological reflectance index was significantly different between the control and the triclopyr treatment 1 day after treatment. This preceded the detection of visual symptoms which were not evident for the triclopyr treatment until 7 days after treatment. One of the most relevant potential applications of this finding in New Zealand forestry would be the pre-visual detection of red needle cast. If this disease could be detected by hyperspectral imagery from, say, a satellite before it appeared and quickly treated, then the impacts on growth may be greatly mitigated. This is further illustrated by a study on olive trees where the aim was to use hyperspectral imagery to detect the disease Oxala fastidosa. The top slide shows the symptomatic trees in dark blue and the asymptomatic trees as unfilled shapes with both categories determined from visual interpretation. The trees that are not coloured in red are those that are not showing any symptoms but were identified as having the disease through molecular analysis. In the bottom image, a model that was developed from hyperspectral and thermal imagery was used to predict whether trees were infected. The key point to take from this image is that all of the trees that were only detected by molecular analysis were also detected by hyperspectral imagery. In summary, using small radiated pine, we've developed strong relationships between hyperspectral indices and photosynthesis and foliage, nitrogen and phosphorus. We plan to scale these predictions up to 4-year-old radiata pine within the accelerated trials over this financial year. We have pre-visually detected the impacts of herbicides on small wildings using hyperspectral imagery. A 3-year-old trial has been established to detect red needle cast from hyperspectral imagery. This could provide a useful stepping stone towards moving towards pre-visual detection of red needle cast in nurseries and the field. Thank you for your attention. I look forward to your questions later.
Tēnā koutou. My name is Loretta Garrett from Scion, and today I'll present on spectroscopy to provide a rapid measure of site fertility in planted forests. The outline of the talk today will start with what is reflectance spectroscopy, what can be predicted for foliage and soil chemical analyses, how spectroscopy has enabled decision-making, and when spectroscopy will be ready for forest industry use. Spectroscopy is all about increasing the volume and flow of data in order to make better management decisions. The technology has been around for a while and is highly used by other industries. It has been successfully applied to soil and crop foliage fertility measures, and is a way of moving away from the wet chemistry laboratory to increase our throughput of samples with accurate and precise predictions. This is the electromagnetic spectrum. You will be familiar with the visible section of the spectrum, which shows up as rainbow colours. Reflectance spectroscopy is focused on the visible to mid-infrared range of the electromagnetic spectrum. The mid-infrared range gives the best detail for soil chemical property predictions and is the focus of our work. The basics of how this works is by shining a light beam onto a sample. This then interacts with all the particles and chemical bonds within the sample. Some light is absorbed and the rest is reflected in all directions, which an instrument detector then measures. Different chemical bonds within the sample will give specific spectral signals, allowing us to know what is in the sample and how much of it is there. How do we go about building soil and foliar chemical property predictions? We start with archived soil and foliar samples from our National Planted Forest Archive and associated chemical analytical data. For example, the percent of carbon within a sample. We then take the sample and measure it on a high throughput infrared instrument, which produces a mid-infrared spectra that is unique to that sample. The spectra is used to predict chemical properties using multivariate analysis, and we can compare how well we do by correlating with our measured analytical data. This work that we are doing is supported by our collaboration with Dr. Jonathan Sandeman, who is an international expert in using soil spectroscopy to predict a number of soil chemical properties. Many soil properties can be predicted well by mid-infrared spectroscopy. This table shows work that our collaborator, Dr. Jonathan Sandeman, has done with the US National Soil Survey Center Mid-Infrared Spectral Library, made up of more than 75,000 soil samples, and they are able to predict over 20 properties from one scan. Tree foliage is less well developed, but shows promise with conifer trees. This example is for a small data set, and the ability to predict chemical properties is good. Then, with any new sample that is collected with no analytical data, we can measure it in a high throughput infrared instrument and predict multiple properties from one spectral scan. We can use spectroscopy to increase the volume and flow of data and build large data sets for any given area for lower cost than traditional analysis methods. This is because with one spectral scan, we can predict multiple properties. We can then make detailed maps to underpin nutritional management zones, for example. The lack of high resolution spatial soil data in particular is a current limitation on informed decision making. Detailed spatial soil and foliar understanding should improve productivity modelling through to forest health and wood quality property predictions. For example, what is the role of soil micronutrients on tree growth, health, and timber products? This level of detail is only made possible with high volume and flow of soil and foliar data, which can be achieved through spectroscopy. So when will spectroscopy testing be ready? By late 2021, next year, the soil and foliar spectral predictions will be ready to use. We have 3 steps to do. We are currently piloting the technology with New Zealand soil and foliar samples. We will then increase the library of soil and foliar samples for improved predictions and then publish the findings. You can support the second step by adding your soil and foliar sample to the library. Ensure that your forest is within the training dataset. If you're interested in this, please connect with me. Ngā mihi.